In the previous video, we discussed the cell-mediated immune response. Now we'll cover the key features of the humoral immune response, which involves B cells and plasma cells, as well as some T helper cells. In the first stage of the humoral response, an antigen can bind to the antibody on the surface of a B cell if it is complementary in shape. Here you can see in red some antibodies on the surface of a B cell with antigens shown in orange triangles. In the second stage, the antigen can enter the B cell by endocytosis, causing the B cell to become an antigen-presenting cell. This is a similar process to phagocytosis, which we covered in a previous video, but it involves a B cell instead of a phagocyte. In the third stage, an activated T helper cell, which was activated by the cell-mediated immune response, binds to the B cell, which is now an antigen-presenting cell, if the receptor on the T helper cell is complementary to the protein on the B cell. The B cell can then undergo mitosis and make several copies. It can also differentiate into plasma cells, which can then produce proteins. These proteins are called antibodies, and they have a very specific shape. They are Y-shaped proteins made from 2 heavy and 2 light chains. This adds up to a total of 4 polypeptide chains. They have a very specific antigen binding site due to a very specific tertiary structure. And once produced, they have the ability to simulate agglutination, which is a process in which pathogens are clumped together and destroyed by phagocytosis. A way to remember this is that the term agglutination comes from the Latin word gluing, and you can remember this by using this word because the pathogens with the antigens are glued together.